Welcome to everyone. Today we are going to explore one of the most fascinating molecular machines inside every living cell. It's called as RNA polymerase. This enzyme is a master builder that converts the language of DNA into RNA. By the end of this session, you will really appreciate how this tiny machine keeps life running. Next, learning objectives, understanding of RNA polymerase, which means what we learned today, first of all, what RNA polymerase are and why they are essential for life, how they initiate, elongate and terminate of transcription, the structural design of these enzymes are truly true molecular marvels, and the difference between the prokaryotic eukaryotic RNA polymerase, and finally we will summarize how all these steps together to make gene expression possible. Mechanism of RNA polymerase. RNA polymerase reads DNA and build RNA, a process called as transcription. The RNA produced then guides protein synthesis, which helps cells alive. Different organisms have different polymerase, but the functions remains universal. The RNA polymerase is essential for the synthesis of DNA, RNA, and the functions during transcription converting the DNA code is into RNA. The structure of RNA polymerase is a large subunit enzyme. RNA is nothing but a subunit enzyme. Like a mini factory, it active sites has two metal ions usually. One is magnesium that from RNA bonds, and the structure is called the bridge of helix, and trigger loop help move along the DNA efficiency. The size of prokaryotic and eukaryotic maybe differs. Prokaryotic polymerase. In bacteria, generally RNA polymerase needs a sigma factor during the start of transcription process. Basically it's called as a promoter. In prokaryotic, which means in bacteria, generally the factor, the promoter region is called as a sigma factor. It helps the enzyme find the correct promoter on DNA. This ensures the correct genes are switched on at the right time. Eukaryotic polymerase, which means eukaryotic polymerase have three main RNA polymerase, that is polymerase 1, polymerase 2 and polymerase 3. Polymerase 1 makes rRNA, polymerase 2 makes mRNA, that is means messenger RNA, and polymerase 3 means tRNA, that is transfer RNA, or polymerase 1 makes rRNA, which means reverse RNA, and the 5S RNA each plays a specific role, specialized role to keep a cellular functions coordinated. The overview of transcription process, it undergoes three different stages called initiation, elongation and termination. Transcription begins when RNA polymerase binds to the promoter region. The DNA unwinds automatically, exposing the template stand, and the first RNA nucleotides are added. During the RNA polymerase binds the promoter region, automatically the DNA unwinds, so RNA polymerase can access the DNA template, which means transcription begins at the site. It's called the overview is called as a initiation of transcription. Second stage is elongation, which means during the elongation RNA polymerase moves along the DNA, adding nucleotides into the 5' to 3' direction. DNA unwinds ahead of the enzyme and rewired behind it, keeps the process smooth, which means RNA polymerase moves along the DNA, adding the RNA nucleotides. RNA is built into 3' to 5' directions using the DNA as a template. DNA unfolded ahead and rewinds begin the enzyme. Finally, termination. When a termination sequence is reached, the DNA strand is released and the enzymes detached. The DNA helix reforms and the newly made RNA is ready for the translation, which means RNA polymerase reaches the terminator sequence, the process will be finished. RNA transcription is released, which means the transcription process will be over. The end product of RNA will be released and the DNA rewinds and the process will be recycled again and again. The enzyme detached from DNA ending transcription. During that transcription is ending over, end product of RNA polymerase is the starting product of translation, that is called the protein synthesis process. The total reactions of converted of DNA into RNA is called as, which means DNA is produced, RNA is called as the transcription process. So finally 3 stages, initiation, elongation and termination will be finished whenever the termination process is over. So finally the translation will be started hereafter. So these are the 3 steps in transcription. The final conclusion, RNA polymerase transcribes DNA into RNA, enabling gene expression. The process, including 3 stages, is called as initiation, elongation and termination. In eukaryotic, different RNA polymerase synthesize various RNA types, which means RNA polymerase transcribes DNA into RNA. This is called as enabler of gene expression, which means transcription will be end and translation will be started. The process including 3 stages, and in eukaryotic different RNA polymerase are synthesized. To be concluded, RNA polymerase defines gene expression by transcribing DNA into RNA. It works 3 key stages and varies between prokaryotic and eukaryotic. Truly, it was a molecular machine that keeps life in motion. So finally, transcription will be over. So thank you for everyone. Thank you, Ananda.